Hi guys, I hope you finished and submitted your first video assignment before you're watching this one. But get ready to take a little bit of notes. You can pause whenever you want, but I'm going to move through this at a normal speed. So we're going to start with a little amino acid sequence review. So 3 bases on RNA make a codon, and 1 codon codes for 1 amino acid. So you have this first codon 5 over here. It's CGG, and that makes 1 codon, and that 1 codon codes for arginine. So that's how it's going to be for every amino acid. But what happens if something happens to one of those bases? What if it changes or it gets taken out? Well, we're going to cover that in just a few minutes. So our first mutation type is going to be point mutations. That's where 1 nucleotide is changed in the sequence. And there's a few different types. So the first type is a silent mutation. That's where the nucleotide is mutated, but that doesn't change which amino acid is coded for. And the next is nonsense mutation, where the codon is mutated to an early stop codon. So if we remember from last time, we learned about how DNA knows when to stop coding for amino acids because of the stop codon. That is, whenever UAG comes up in a chain, that's where it stops coding for amino acids and leaves, just flies away. So then we have a missense mutation, which also is kind of the opposite of a silent mutation. It's where something is changed, but then it causes a change in the amino acid chain, whereas the silent mutation, there is no change in the amino acid chain. Our next mutation type is a frame shift mutation. So that's when a number of nucleotides causes a change in the way a strand is read. So there's a couple different types of this as well. The first is going to be an insertion. That's where a group of nucleotides is added into the sequence and it disrupts the triplet pattern. So if you remember, our codons are groups of 3, so that's the triplet pattern. If something happens where 2 amino acids come in the middle of one of these codons, or even after one codon, it's going to cause a push, and then it's going to redesign the way the genetic sequence is read by the amino acid coder. So that's going to change the amino acid sequence from the point of insertion on. And something similar yet opposite is a deletion. That's a group of nucleotides being removed from the sequence that disrupts the triplet pattern. So if you have 2 codons right next to each other, and these 2 get taken out, then this one now it takes 2 from the next codon and then redesigns the amino acid sequence after that. Crazy, right? So just to make this a little bit more real for you, some examples of mutations in the genetic code in real life. We have sickle cell anemia, we have Duchenne's muscular dystrophy, and cystic fibrosis. And for the frame shift mutations, we have Tay-Sachs disease as well as Crohn's disease. Some of you may have heard of these before, some of you may haven't, and that's okay, because we're going to cover them a little bit more later. Now, I emailed on our Canvas site, our Moodle site actually, under this week's unit, so you should have a little tab that says mutations. I have a chart that I need you to either print out or redraw and cut out on your own, where you're going to cut out all of the nucleotides and amino acids, and you're going to build a small model of DNA. And this is going to be helpful for you to work out some mutations later and actually visualize what happens. So that's why you have so many little nucleotides to cut out, and then you have your DNA, RNA backbone to put them on later. So you can go ahead and cut that out on your own time. Make sure you get something like an envelope or a Tupperware to keep all the little pieces in. I know they're kind of small, but you're going to be using that in the next week or so